Hello, I'm David Pompliano, the CEO of BioLeap. At BioLeap, we do alternative drug discovery, or as I like to say, we design smart molecules. I'd like to tell you a little about our approach. You may find that BioLeap can be a vital partner in your own work. Everyone in the pharmaceutical industry knows that drug discovery often leads to dead ends. The work is fiendishly difficult. Yet discovery needs to be more productive, especially given the challenges that now confront the entire industry. Amid these challenges, BioLeap works with partners towards several ends. First, to speed discovery and optimization of drug leads. Let's shorten the path of development. To moderate costs, let's limit compound synthesis and testing to those with the highest likelihood of success. To create novel intellectual property or to develop a fast follower, let's increase the chemical diversity. And ultimately, to deliver usefully novel therapies, let's improve healthcare. What we offer is a step change in molecular design technology and a team that knows how to exploit it. We are advancing computational, fragment-based drug design to an unprecedented level of predictive power. The power to rank the odds of success for novel compound designs before synthesis and lab testing. A high degree of predictability is essential to an effective design process. Now, as you might expect, our team includes people with many years of experience in refining fragment-based design. Crucially, however, the team also understands the difficulties of medicinal chemistry as it's practiced in the lab. John Culp, one of our founders, has worked in BioLeap's core technologies for 12 years. He is the inventor of BioLeap's many advances in predicting the binding of fragments to a target. Rick Bryan, another founder, has worked with John for these past 12 years, and he is the architect of BioLeap's proprietary fragment-based design tools. Ian Cloudsdale, our head of chemistry, has more than 2 decades of experience in small molecule discovery. He is the co-inventor on 24 US patents. I myself am a chemist who has had the good fortune at large drug companies to work on what are now 4 marketed products. Yet, like any drug hunter, I've witnessed failures too. In one 7-year effort to discover antibiotics by high-throughput screening, more than 70 targets were screened against as many as 500,000 compounds. The screening facility was state-of-the-art. Yet that campaign did not find a single compound that progressed to be a drug candidate. There has got to be another way, and there is. At BioLeap, we do not rely on chance or guesswork. We do not test random assortments of existing compounds hoping for a hit on the target of interest. Instead, we determine which small chemical building blocks, fragments, the target wants to bind, where it wants to bind them, and with what affinity. Our advantage starts with our computational algorithms. Using rigorous thermodynamic principles, we create a fragment map of the target. Then our chemists design small molecules to bind to the target with high potency and selectivity. Throughout this process, we are guided by our proprietary data and design tools. As we assemble fragments into a ligand, we assess the effect of linkages on binding, making adjustments as we progress, fragment by fragment, substitution by substitution. Finally, we rank order the completed ligands by affinity so that only the best few ligands need to be synthesized. This is truly design de novo, and it is nothing like trying to force fit an existing compound into a target. I'd like to emphasize several points. One is chemical diversity. The theoretical number of possible fragment assemblies, that is drug leads, is in the trillions. This diversity serves not only potency, but also sets essentials as bioavailability and ease of synthesis. What's more, the process of assembling these fragments one by one can itself reveal non-obvious linkages leading to novel structures. Now, another point concerns the significance of water in drug discovery. Water naturally bound to a target will of course affect binding of other molecules. We can predict water binding just as we predict fragment binding, even when waters are embedded and may not be detected by any other methods. So, we can prevent water from confounding our design, or we can use it to our advantage. Lastly, I'd like to come back to the predictive ranking. Our confidence in this predictive power is supported by our validation studies. In one example, in a blinded study for a major pharmaceutical company, we predicted the order of compound affinities with greater than 80% accuracy, far exceeding the capabilities of other in silico methods. Our process goes through several design cycles on the way to a development candidate, yet the compounds synthesized through all cycles will usually total fewer than 200, or about 1/10 what you would expect in a traditional drug discovery program. Hence, the savings, not only in the budget, but also in the time period needed to achieve preclinical milestones, is significant. We will collaborate to create lead compounds or partner to produce development candidates. In either case, the goal is a smart molecule, not silly molecules that can make drug discovery campaigns long, expensive, and futile. Have a look at the other features on our website. The videos demonstrate our capabilities in much greater detail. I think you'll find that BioLeap offers a genuine alternative in drug discovery, an alternative that will give you a competitive advantage.